handful of speakers. We are going to try and wrap up by 11:30 because there's another meeting of the ISO team to follow in person that starts at noon. So for those of you who are joining that, we will try to give you a little bit of a break between this meeting and that. So we're aiming for 11:30, but we'll have some time for open discussion. Sure. We are going to be recording this meeting, so please let me know if anybody has any issues with that. And give me 1 sec to share screen and we'll get started. For those of you who haven't seen, on the Open Oximetry website, the recordings from prior meetings are there. This one will also be posted there. The minutes from prior meetings are also on the site on the community page, which can be accessed at that link and that QR code. This is the rough agenda that we'll try to stick to as best we can. Sorry for the typo at the end. We'll wrap up at, try and wrap up close to 11:30 or 11:45. We'll just do a quick recap of the March meeting and what's been happening since then, and then we'll get some updates from the ISO and FDA teams on upcoming regulatory changes and plans. We'll have a little bit of discussion, a couple of presentations with some new data around some skin color quantification methods that have been a hot topic in this group, and a lot of people working in that space. And then we'll share some updates from some of the prospective studies happening in a couple of sites through the FDA CERC centers, and then we'll wrap up. Again, just a reminder, please keep yourselves on mute. If you have questions, please use the raise hand function or chat in the chat here, and we'll try and call on you. We're happy to moderate some discussion after the presentations, but again, this meeting will be briefer than last time, so we may need to continue that. And just a reminder for those in the room, leave the audio if you haven't already. For those of you who don't use Zoom very frequently, this is how you can raise your hand if you do want to be called on. Before we get started, I just want to acknowledge the team here in San Francisco that's done a lot to help put this meeting on and host the afternoon meeting, also conducting a lot of the work. Special thanks to Fifi Nuguse, who's partnering on a lot of the work with Open Oximetry and planning these meetings as well. Also, a special thanks to our speakers. So while we're here, this is sorry, we're just troubleshooting some audio stuff. Mike, why don't you just disconnect from Zoom? Okay, all right. So the reason we're here, obviously, I always care a lot about patient safety fundamentally, as well as health equity. The disparities that were highlighted as a result of pulse oximeter performance have kind of rekindled some interest in trying to improve standards and guidelines to try and address some of these things. And so today, part of the purpose of this meeting is just to share updates on what's going on. It's been 6 months since a larger group has met, but there's a lot of work that's been happening in between that many of us touch pieces of. And so we're hoping that we can promote some cross-pollination and communication from a diverse group of stakeholders in that. Most of us are already well familiar, but just a quick recap. Part of the backstory leading up to this has been data from the 80s, 90s, 2000s that have been mixed but concerning around this issue of performance variability of pulse oximeters, especially in patients with darker skin color. But it really wasn't until 2020 during COVID where there were health and healthcare disparities that were linked to this performance issue. A lot of initiatives, multiple communications from the FDA and other national regulatory bodies in many countries to try and start addressing these issues. FDA had an open public hearing in November of 2022, and we had a forum in 2023. And then there have been, again, a number of side meetings, and obviously the ISO committee has been extremely active in trying to address many of these issues. I'm not going to go too much into the March meeting. We very broadly discussed, tried to nail down some problems and potential solutions that could provide a research agenda for many of the teams. And again, that meeting is the recordings by session are on that page. If you haven't seen them and want to catch up on those, you're welcome to check those out. After the March meeting, we did form a handful of subgroups that were focused on different topics. as much as possible, we have tried not to duplicate what the ISO committee has been doing and tried to join those meetings. But there have been a few groups that have been meeting, in particular the the clinical trials subgroup. There are a number of prospective clinical trials that are ongoing around the world. And so that group has met a handful of times. There's another group working on an open data repository. We'll hear a little bit more about that. And then two groups that more or less have been overlapping with the ISO groups around some issues around raw signal and skin color quantification. If you're interested in joining those groups, I'll give you a little bit more information about those. Also, the OpenOx project, we did accept applications for individuals, companies, researchers, anybody who was working on devices that they wanted to do controlled breathe down studies to test their devices to see if they had come up with solutions that address this issue of variable performance related to skin color. These are 3 of the teams that were selected. We appreciate everybody who applied. We weren't able to accommodate everybody, but these are 3 of those solutions. And we'll be testing these devices hopefully end of 2023 or early 2024, and asking those groups to come and share their findings in one of these meetings coming up. So stay tuned for that. And thanks again for everybody who applied, and congrats to those who were awarded those. At the end of today, we'll have the updates from the real-world clinical studies. There are a handful that are on, there are multiple that are ongoing. We'll hear from 2 today. And for those who are on the line, part of the clinical trial subgroup, if anybody wants to share brief updates, at the end of the agenda today will be the time for that. We have published on the website 6 study protocols that mostly deal with improvements in breathe down studies, protocols that we've shared for prospective clinical trial data collection to try and standardize data collection processes. Also protocols around skin color assessment, and have appreciated everybody's input. Many people in this room and online have been really helpful in contributing to those, but those remain a work in progress and are constantly open for ongoing comment and feedback through the website. So we appreciate anybody sharing on those, and hope that one of the outcomes of today and the afternoon meetings will be potentially some amendments to some of those protocols. Also of note, we have been exploring with collaborators and partners in Uganda the possibility of opening a hypoxia lab. Getting an echo from somebody online. Just a reminder for everybody to mute. We are, UCSF has a longstanding partnership with multiple anesthesia and critical care departments in Uganda. Some of our collaborators there are part of our Open Oximetry core team. We'll be hosting a stakeholder meeting in Uganda. PP and I will be there in a couple of weeks to talk about the possibility of opening a hypoxia lab in Uganda to try and improve the diversity of data that is collected and representation of other patient populations that aren't always accounted for completely with the current device testing labs that are limited, as we all know, and predominantly in high-income country settings with populations that could be more diverse. So please stay tuned for that. We hope to report back in our next meeting, findings from that meeting. As for this forum, I'll kind of wrap up with this. Again, we are hoping that this forum can continue to meet at least a couple times a year, if not more frequently, to share updates and promote communication across the different teams. And our intention is still to create a new collaborative community as described by the US FDA, but to be a working group that really can work on a number of things that are outlined in this next slide. This includes trying to build consensus around best practices, SOPs for data collection and analysis. Again, obviously a lot of that is really being led by ISO and other regulatory agencies. We just want to be able to work with those teams and try to bring other input to the table and also share data to inform those in as real time as possible. We also are excited to try and work on some communication and education material, both for clinicians as well as for patients. And I think that would be a great role that the forum could play that sometimes is a bit out of scope for a regulatory agency, for example, or but we think that's something that the collaborative community could be very uniquely positioned to be able to do well. Advocacy and not just for patient stakeholders from resource-denied communities, but also making sure that we're continuing as a group today to advocate for diversifying this group, diversifying regulatory agencies, diversifying all of these entities that would benefit from having more stakeholders at the table and more voices. And community engagement is another piece of that we're hoping that Open Oximetry project can contribute through this forum to the broader discussion on what we are all working on. So if you haven't, you can join the collaborative community, the forum communications through the Open Oximetry website. There's a button on the page. You enter your email, you just get on a listserv. We rarely send out emails, so this will not be like a common source of spam for you. So please sign up if you haven't already there. So that I will end there. And at this point, I'm going to hand it over to Dr. Liz Igaga to introduce our speakers from ISO and FDA, who are going to provide some updates on where things are with regulatory changes in the pipeline. Dr. Igaga, many of you have met in the last meeting, is an anesthesiologist based in Uganda, a patient safety advocate, completed a patient safety fellowship, has worked with multiple nonprofit groups, including Smile Train and Lightbox, and is a real advocate for patient safety in East Africa and beyond. So thanks, Liz, for joining. And with that, I'll hand it over to you to introduce the next panelists.
All right, thanks, Mike. Please let me know if you can hear me.
Sounds great.
Yes, I'd like to have the honor of introducing Dr. Edelman Malvina, who is from the FDA. She is currently the Director of Office of Ophthalmic Anesthesia, Respiratory, ENT, and Dental Devices at the US FDA. She received her MD from Harvard Medical School and a doctorate in health sciences and technology from MIT. And for over 20 years, she has worked as an expert medical officer, a senior medical advisor, the director of the Division of Ophthalmic, Neurological, ENT Devices, and director of FDA's Pilot Office for Ophthalmic, Dental, Respiratory, ENT, and Anesthesia and Sleep Devices. She has also played a key role in assuring the safety and effectiveness of medical devices. So Dr. Edelman guided the development of more than 50 international and national standards. She's overseen development of numerous regulations and guidance and has convened over 30 public meetings of the FDA Medical Device Committee. So it's my absolute pleasure to invite her to come and give her presentation. Welcome.
Thank you very much for that very kind introduction. Let me try to share my slides, and please let me know if you can see them.
We can see.
Perfect. Okay. So thank you again for having FDA as part of this incredibly important forum. It has been our absolute pleasure to participate and learn from all of you. I am a strong proponent of collaboration to expedite innovation in every important area, and none is as important to us as assuring health equity for all patients around the globe. So I'm gonna do.
Sorry to interrupt. Can you make your slides full screen? We're seeing just to make it a little easier to see.
Okay, hold on a second. How about now? Is it better?
Perfect. Thank you.
Okay, thank you for letting me know. Okay, I'm gonna go through this pretty fast because I know all of you have before, but for those who, okay, my Zoom is acting funny, so if you lose me, let me know somehow. I don't know what's going on. It's okay, don't send. Can
I think we lost her for a minute. Dr. Malvina, your screen went off and you're muted.
Okay, for some reason, my firewall kicked me out. Let me know if you can see and hear me now.
We can see your screen and we can hear you.
All right, try 2. Hopefully there won't be try 3. All right, so all of the pulse oximeters in the United States can be basically categorized in two major buckets: those that are prescription use pulse oximeters and those that are over-the-counter, or as we refer to them, OTC. So the prescription use pulse oximeters are reviewed by FDA through something called 510(k) pathway, which means that we ask for new devices to show substantial equivalence. And all of them undergo clinical testing to confirm their accuracy. We do have a recognized consensus standard, which all of you know and will be hearing about very soon. And in 2013, we issued a pulse oximeter guidance, which I'm going to be summarizing some key aspects of very soon and very fast. The OTC pulse oximeters are most commonly intended for general wellness or shortage aviation users. These are obviously sold directly to consumers and online. And up to now, we have been utilizing our guidance for general wellness and policy for low-risk devices to regulate these. Okay, let me see if it'll let me go to the next slide. Okay, here we go. Do you see the next slide? Good, thank you. All right, all of our 2013 guidance recommends that all of the new pulse oximeters and all pulse oximeters with significant modifications perform in vivo testing prior to them reaching the market. And the purpose is basically to verify that the accuracy is comparable and adequate for our needs. Okay.
Go ahead now, Julian, sorry.
Okay, thanks. Does that include OTC that you just mentioned? The testing, the clinical testing?
Not at the moment. Did I stop sharing my screen again? I am sorry, I don't know what's happening with my. Okay.
And I'd ask that we keep our questions until the end, just so that we can get through the presentations, and then we'll have some time for question and answer at the end.
Okay, thank you. Okay, 2013 guidance does talk about the number of subjects, and it's a minimum of 200 data pairs for our assessment. Now, it's also important to note that the guidance recommends that we include Okay. I am not sure what's happening with my slides. Perhaps I can. Did I lose the slideshow again?
Yes, you did. Can I suggest that we go through the other presentations first?
Yes, please. Perhaps, can you send me an email and I'll reply with my slides because it keeps kicking me out.
All right.
Dr. Edelman, if you want to send them directly to me, I
will do that. Please go to the next one. I'm having trouble with my Zoom this morning.
All right. Sorry, guys, about that. While we try to figure out the technical glitch, I hope that Sandy and Bob are ready for their presentation. We will go straight to them. And again, I am pleased I have the honor of introducing Bob Kokotic, who is from the ISO. He is a dual licensed clinician, a registered nurse, and a respiratory therapist with advanced degrees in bioengineering, critical care, and physiology. And he's currently employed by Edwards Life Sciences. And while Edwards does not manufacture a pulse oximeter, Bob's prior industry employers included 4 pulse oximeter manufacturers. Regarding international standards, Bob is a co-convener for the IEC ISO device standards related to oximeters and the subject matter expert to AAMI, IEC, and ISO on blood pressure, critical care, and ventilatory support devices and cybersecurity as applied to medical devices. Bob is a member of the Sigma Theta Tau International Nursing Honor Society, and a distinguished fellow of the American Association for Respiratory Care, and also a fellow of the Association for the Advancement of Medical Instrumentation. And he is going to present alongside Dr. Sandy Weininger, who is a senior engineer working at the US FDA Division of Biomedical Physics of the Office of Science and Engineering Labs within the FDA Center for Devices and Radiological Health. His current scientific and regulatory focus is the safety and performance assessment of sensors and actuators for physiologic systems and examining complex systems to understand unsafe interactions. He works with standards development organizations such as UL, AAMI, IEC, and ISO to construct both horizontal and vertical safety standards. He is a member of AAMI UL 2800 interoperable systems, a co-chair of AAMI's interoperability work group, a co-chair of JWG10 of ISO, oximeters, and one of FDA's liaison to IEC and IEC TC62 and SC62A committees responsible for safety of electromedical equipment. Dr. Weininger works with academic partners to develop methods for safety and performance evaluation of interoperable systems. And with that, I will hand it to Bob and Sandy.
Very good. Can you hear me?
We can hear you. If you could put your slides in slideshow, that would be great. And now we can see your slideshow. Thanks.
There now. I was reticent until I knew you were winding down. So sorry for the long introductions. Yes, Sandy and I have worked together for several decades in many forms, but primarily with the focus on oximetry devices. And there are several of them. We're focusing this meeting on one of them, and that's pulse oximetry. And what I wish to point out here in this quick scan of individuals and what's going on with happen in this particular clinical setting is look at each of these clinicians. None of them are looking at the patient monitors in the background. I'm not saying that monitors aren't important. I have used them clinically and continue to do so in my laboratory and other efforts. However, these monitors are adjunctive. They're meant to assist in the assessment of a patient. So as such, they are not the end all, nor even a direct measurement of the value that we are trying to assess in looking at the patient and attempting to stabilize them in either an acute setting like this or in their chronic disease. So I wanted to make sure that that point got across because pulse oximeters, whilst extremely helpful, are an adjunctive monitor. Now, these devices are doing what is representative of what's happening inside the patient. This is the brain, these are the feet or other peripheral sites, and these are values of direct blood oxygenation measurements throughout the body. And I only point this out because the body is actively consuming as well as receiving oxygen. And it's a direct balance that you're trying to ultimately make sure that these patients are receiving enough that they are not having problems. What are the indications of problems? Well, lots of additional disease. And that means kidney failure. It means brain oxygen is disadvantaged to the point where mentation is impacted and potentially even to the point where someone may be fatally injured. So SpO2 can be very helpful in assessing things like perfusion, but only as an indirect measure. So I bring that up because I want to make sure the audience appreciates why this indirect measure is really challenged by what happens with what some people have referred to as physio-optics, meaning there's physiology and there's light and the method of being able to detect that light. These are two light sources. One is red, and if you've seen a pulse oximeter in operation, that is the typical color you see as it's glowing over a finger or a nail bed. The other color is infrared, which is not visible to the human eye, but yet is there and is easily detected by this special detector. That optical detector is challenged by the tissues that are in between. And the tissue of greatest interest are these circulating red blood cells that are picking up oxygen and delivering it to the tissues that need to metabolize that oxygen. And as you note, as this light is emitted in a constant way, a constant power, that light is reduced or absorbed or attenuated because of the color of that blood. So that's what we're talking about, is where are the challenges to this technology? They are motion, because they can fool what is assumed to be the arterial pulse, which is where that detection is focused upon. It's low pressure, as well as other interfering substances that are colored but may match or confuse the color that we're trying to get to in red blood cells. This is one such example. And I kind of wish that I had made this graphic such that what you then see are only the nail beds. These are oxygenated nail beds, but you notice that they're rather white. The fingertips are being pushed down. If you look at your own fingers as you push them down on the table, you notice they blanch or they reduce that color. Whereas these fingertips are actually detecting or showing cyanosis. They're blue. And that's particularly challenging in dimly lit clinical conditions, as well as in home care settings. So having a device in place that can correctly detect, regardless of skin color or other color concerns, is vital to making sure that there is continuous assessment of this oxygenated hemoglobin. That the point at which cyanosis can be seen is actually almost 30% lower than what is normal oxygen saturation. So this is what is challenged as we try to detect unoxygenated or lower oxygenated red blood cells versus oxygenated. And we can only focus upon this arterial pulse. So all of this other blood contained within venous or capillary blood, or captured within tissues, or the tissue colors themselves, have to be ignored. And yet all of this represents light absorptions. So how much is left over once you throw that light in? Less than 90% of all of the light that goes in from that finger sensor you see glowing red is actually able to be detected, and then we focus upon only the pulsing portion. So when the signal is the smallest, we're attempting to derive that oxygenation value. Now, I only point to this slide because people may not be familiar with, you need to get these plateaus of desaturation. That is all well defined in the standard, and even more so, all of the safety concerns of that, from a publication here of just a few years ago amongst multiple sites that were doing studies, including obviously Mike Lipnick here is credited for this as well. This is the kind of very stable, controlled setting in which that desaturation is occurring. I just wanted to emphasize how it happens, but more importantly, what happens in the real world. This is a sample of over 32,000 paired data sets in real world testing of babies. Now, this was 20 years ago. What I want to point out is their closing statement, and as well the activities that Sandy and I have been engaged in over that same period of time that address this matter. So these are 5 different pulse oximeters, and they said while the accuracy was device dependent, meaning they differed between them in the range of interest, 92 to 97, above and below that range, they varied dramatically, and so dramatically that they gave an example. At 80% SpO2, now remember, cyanosis shows up at about 75% SpO2. The real co-oximeter, the real blood value for oxygen, was 10% higher than that. Now, this is 20 years ago. I just wanted to be certain that you understand that some of these technical challenges of deriving when the signal is the smallest, manufacturers have been able to dramatically improve, but in a controlled laboratory setting, which is, as Malvina pointed out, is the result of this ISO standard as well as FDA regulations in domestically. Now, these regulations for ISO are cornered by other IEC as well as ISO quality systems, as well as risk management and the supportive standards that make sure that these devices are immune to other sources of contamination, such as radio waves or light or various other challenges. So there's a lot of safety issues built into these pulse oximeters that may not be appreciated unless you're looking at these manufacturer-based standards. These are the standards that a device manufacturer must adhere to in order to release their product. The last such standard was in 2017, and there was a minor update to that just a couple of months thereafter. And as you know, it's not only basic safety, what is intended by essential performance is also accuracy performance. And so that's what I will be focused upon here, because with that accuracy performance comes enhancements to the standard that I just pointed out that is now going back to 2017. So here we are with the committee draft stage that went out just about a month ago to all of the manufacturers that are part of the 89 countries that are active within ISO and IEC. And those updated manufacturer expectations come with various challenges too, and they are to comment upon this new version of the standard, which is still a draft. And I want to emphasize that it has been only distributed for review and comment by those 89 countries, all of which have representation from their device manufacturers, but it could also include clinicians from those various countries. We are thankful, Sandy and I, for some better than 40 active participants on our committee that represent the world of manufacturers as well as specialty clinicians. Sandy, I'm going to pass this over to you, and if we have time, the final would be a review of what the rationale of why we undertook that. Sandy, are you unmuted?
I am unmuted. Can you hear me?
Yes.
So I will go through this relatively quickly. Dr. Edelman unfortunately wasn't able to show what was in the pulse ox guidance because a lot of that is the basis of what we have in our standard. We try very hard to harmonize with the FDA guidance document so that we have one common safety and performance standard, and that way we can harmonize and bring markets and innovation, bring devices to market and innovation more effectively, we hope at least. So I'll just highlight some of the things that have arisen within the last 2 or 3 years that we've been discussing. And FDA is considering some of these changes too in the new guidance document. The expected levels of performance are going to be tightened up. The current version of the standard says your oximeter should perform better than 4%. That's going to be tightened up to better than 3%. There's some controversy around that because it depends on the kind of oximeter that you're looking at. Some of those over-the-counter devices that Dr. Edelman referred to may or may not get to 3%, and we could have a discussion about availability and cost versus performance because you can't have both. All right. There's a much heightened sense of trying to assure that we have diversity and equity built into assessing the performance of oximeters, and we're working hard to figure out what that means. Same caveats apply that when you ratchet up the evaluation and acceptance criteria, a lot of times many of the devices are not going to pass, and so you might lose some availability. And so we have to figure out, are those not going to be allowed to be marketed? Are they going to have special labels on them? What are we actually going to do with such things? Next, number 3, the sample size was at least doubled, and the criteria for selecting subjects was greatly improved to assure that we have that diversity and equity in the patient population that we're representing. And that's 3 and 4. Next slide, please, Bob. We worked very hard with different methods to assess from a perceptual, I was going to say perception, sorry. Anyway, from a perception, which subjects should be included, and then trying to make objective measurements from those. The data really is still coming in as we speak. And in fact, the UCSF folks are doing a lot of the heavy lifting to try to understand what the data looks like. So at least in this version of the guidance document, or I'm sorry, in the standard, we're specifying what our best guess is. And we expect as more data comes in over the next year to tune this up.
Okay, Sandy, the rationale section was too large to break out, given that we have 5 minutes.
And I think we indicated. I mean, Bob indicated very well. There's a lot of different clinical applications ranging high acuity to low acuity. Certainly, COVID highlighted the need, perhaps, for oximeters in much broader distribution. And again, we can have that discussion about whether your oximeters should cost $500 or $1,000 and have 2% uncertainty, or they should cost maybe $25 or $50 and perhaps only have 3 or 4% uncertainty because you're using them more as a trend device, right? So those are the kinds of questions which don't, if you will, have absolute answers, that require the judgment and perspective of all our committee members. And so let me say just a brief word about that. Bob alluded to the makeup and the nature of our committees. Our committees are made up of manufacturers and clinicians and regulators and academics and anybody who wants to participate. All members of ISO, all ISO participating members can and should participate through their local national committees. So for example, in the United States, AMI, the Association for the Advancement of Medical Instrumentation, administers the United States Technical Activity Group, the US TAG. And that group is responsible for collecting and coming up with a consensus opinion about what comments should be submitted to ISO, in this case, well, ISO IEC, but I'm using it shorthand for ISO because it's an ISO-led project. You know, what the vote should be on the different, when the votes come out, and what the comments submitted through. Each country has the same thing. There's another mechanism that we have, and we can establish formal liaisons between different organizations, and those liaison agencies can have access to all our documents and can comment, but they can't vote on the particular standard. So there's lots of ways to get input into the standard. Bob and I have been accepting lots of participation, but we've kind of been asked to clean up our roster a little bit, and so we're going to have to be a little bit more careful with the status of each individual, because it's not fair to the members who do participate to just keep accepting new people in all the time. So
Sandy, well done. I just got a message. We are 2 minutes away from our end time, so I think we can wrap it here. I am not trying to show us this slide as an excuse of why we are where we are, rather to emphasize the challenges, the technical challenges, as well as the clinical challenges of making a device that needs a lot of clinical education and end user education if it's being used in the lay group of individuals, meaning patients themselves or just interested people in their well-being having such devices. So we are putting criteria around a lot of that to make sure that manufacturers are doing better declaration of how to make the device work best on yourself or on a patient, and to ramp up the expectations that these devices are performing as best as they possibly can, even though they are indirect measures of arterial oxygenation. All right, so the baton is back.
One final word, Bob. I thought you were just showing this slide to show that our heart rates are elevated, and so anyway, go ahead.
Well done, Sandy. Yeah, we'll chat this afternoon. Very good. I'm going to stop sharing.
All right. Thank you very much, Sandy and Bob. That was very enlightening. We are going to go back to Dr. Malvina, who I believe now, Mike, are you going to share her slides?
Yeah, and I'm going to keep my video off. Hopefully this will keep me on Zoom and not kick me out.
All right. So once we're done with that, then we will have questions.
Yeah, then I'll turn my video back on. Okay, so, thank you, Sandy and Bob, for that presentation. A cornerstone of our guidance document and review is the use of the internationally recognized standards. This standard establishes the basic safety testing needed and defines protocols and tests that are utilized in the evaluation of pulse oximeters, defined performance. But again, I just want to re-emphasize that the modifications that Sandy and Bob just presented have not been finalized or accepted by the ISO yet, and nor has FDA recognized an updated standard. So with that, we're going to go back to what is in place as of today. So I believe I stopped here before. The 2013 guidance does recommend in vivo testing for SpO2 accuracy for all new pulse oximeters and all prior cleared pulse oxes with modifications. Next, please. I'll try to go very fast. The guidance does provide recommendations for pooled 200 data pairs, evenly distributed over the tested range. And again, the guidance makes recommendations for maximizing physical characteristics, but at the moment it only asks for at least 2 darkly pigmented subjects or 15% of subject pool, whichever is greater. Next. You heard a lot about the standards, so we can skip the slide. Next. To verify SpO2 accuracy, the 2013 guidance recommends comparisons to the gold standard measurement of blood SaO2 by a co-oximeter over the entire tested range. Now, again, I want to emphasize that as of right now, it is based on pooled accuracy ARMS across the entire tested range, and there is no current threshold for accuracy analysis, and the analyses are not currently powered to determine significant differences between cohorts based on age, sex, or specific pigmentation values. Next. So all of you probably know all of this, but I'll just summarize it for those who might not. We all know that the scientific community has been aware of the impact the accuracy that certain patient factors have on the accuracy of pulse oximeters, and one of these being skin pigmentation. And I've alluded to what we recommended in 2013 FDA guidance regarding pre-market studies and addressing this. Now, between 2013 and 20, there was some limited literature on the subject, and obviously the Shorting article in 2020 reported that Black ICU patients had approximately 3 times the rate of occult hypoxemia compared to the white patients. And the article concluded that reliance on pulse oximetry to triage patients and adjust supplemental oxygen levels may place Black patients at increased risk for hypoxemia. This started a lot of media attention and has brought it to the spotlight for all at FDA. We have reviewed the literature at the time, and we wanted to provide the public with information on the interpretation and limitations of pulse oximetry. And therefore, in February of 21, we released a safety communications with, go back please, with recommendations for providers and patients. Previous slide, please. At that time, the FDA committed to the combined evaluation of safety and effectiveness of pulse oximetry. Then on November 1 of 2022, we convened a virtual public meeting of the CDRH Anesthesiology and Respiratory Therapy Devices panel to share information and perspective from all of the interested parties. Now, next slide, please. We wanted to discuss the impact of skin pigmentation on clinical performance. We wanted to obtain recommendations regarding validation studies so that we can update our pre-market assessment. And we also wanted to publicly share what we thought, our views on the issues, and provide a platform for all of the other important stakeholders to express their views. It was a very impactful meeting. And while I can spend an hour summarizing all of the discussions, the shortest way I can summarize panel's recommendations is to say that there was definitive consensus that demographic factors, form, fit, function discrepancies, and perfusions need to be addressed, that subjective and objective measurements should be obtained with equal representation of race and pigmentation. There was a recommendation to tighten arms to 1.5 to 2%, and also there was a recommendation for more clinically meaningful accuracy metric. In addition, there was a significant discussion and concern over inaccurate and unverified performance of OTC pulse oximeters. And most panel speakers and panel members recommended FDA come up with a better, clearer labeling about the OTC performance. Next, please. So since the panel a very large team at FDA has been struggling with a key issue, which is how should the pre-market desaturation studies be designed to better answer the reported disparate performance in the real world. And the reason we needed a big team is because we wanted to make sure that we incorporate all available data sources. We looked at the pre-market pulse oximetry data that we have in-house. We have combed through a fine-tooth comb the advisory committee meeting and looked at the different feedback provided by different stakeholders, including patients, clinicians, researchers, academicians, and industry. We are, extremely grateful to be able to be part of 2 real-world studies at UCSF and Stanford. And we have continued learning from exchanging scientific ideas and research, both from participation at ISO, that Sandy so eloquently summarized, and as well as this group, which has been truly an incredible source of new ideas, new concepts, and we're very grateful for the work that all of you are doing. Next slide, please. So again, it was a lot of issues that we're trying to discern, but I tried to summarize the key issues in 2 slides. So one would be, how can we assure non-disparate performance? And the key questions that we're trying to answer are, what are the limitations of current technology? What qualifies as clinically relevant bias? What are the best predictors of disparate bias? What is a feasible statistical analysis plan? And what is a minimum sample size to show statistical significance? Next. The next big bucket, the most important bucket, I would say, is all of the considerations that have to do with pigmentations, race, and ethnicity. We want to assure inclusive and equitable representation, and we're trying to identify best methods to capture race and ethnicity diversity in pigmentation. We're trying to establish the most reliable and accurate subjective and objective pigmentation assessments, while trying to identify the best anatomical locations to capture pigmentation relevant for the pulse oximeter performance. Next. So, as I said, we have been doing a lot of work since not just since the last panel meeting, since we identified this as a significant issue. We are currently considering multiple approaches to address clinical performance issues based on currently available information. And we anticipate seeking additional public input on our current thinking. And when we do, I hope that each of you takes the time to share your thoughts, because we actually read every single comment we receive. We want to make sure that the direction we choose is best for all parties, for every entity in the medical device ecosystem. And that concludes my presentation. If you stop sharing my slide, I'll try to turn on my camera and hope that I don't get kicked out of Zoom.
Thank you, Dr. Malvina. I had posted in the chat that if you have any questions, it would be great for you to post them in the chat. I see Sandy has already answered one of the questions. For you, Dr. Malvina, there's one question. Julianne Goldman is asking, can you clarify if OTC designation will start requiring in vitro data? What is ProCode?
Okay, so that is going to be one of the things that we share for public comment. We're in the process of finalizing our thoughts, our proposed recommendations, and we will share our thoughts via a number of avenues. And at that point in time, we will ask for your input. At this point, I cannot say definitively which way we're going to go.
All right. I'm just going to, one last question.
I-
Francois
Can I,
says
make sure- I don't know how the system works. Can I just ask should I mute mine?
I just muted mine. I thought you wanted me to ask in stereo. The follow-up question is, we've discussed this in the past, and I'm wondering if this applies. The pathway for non-invasive blood pressure monitoring enables consumer access to the devices. So they're sold for non-prescription use, but they are clinically validated devices. I don't know how that maps into your thinking with regard to OTC, but essentially that's the case with NIVP. They're sold OTC, but they all can require clinical testing or in vivo testing. So could you comment on whether that model will just become consistent along with these devices?
So Dr. Goldman, we have a number of OTC devices, and each has a slightly different paradigm. In deciding what to do with OTC pulse oximeters, we are looking across all different ways that we regulate OTC devices to try to come up with the best possible way to accomplish our goal. And we will be once again communicating what it is very soon. And I hope that you'll be able to comment and share your thoughts once you see our proposal.
All right.
These products
Thank
during.
you very much. My apologies. We have one of our speakers who needs to leave, and so we will take questions after that. Please continue the conversation in the chat. I think it's going well, but I am going to hand it over to Mike.
All right, thanks so much, Liz. So we'll move on to the next session. So I think many of you know me, but just by way of introduction, I'm Fifi Nuguse. I'm a program manager for the Open Oximetry project. Specifically, I've been co-leading our efforts to diversify our studies and make our research practices more equitable. I have the pleasure today of introducing our next speaker, Dr. Leo Smiljovic, who will be presenting on optimized skin color quantification techniques. Dr. Smiljovic is an assistant professor of medicine, radiology, physics, and biomedical engineering in the Division of Dermatology at Washington University of St. Louis. His research interests include developing low-cost optical imaging devices that are globally accessible and developing methods to quantify skin mechanical and optical properties and interrogating disease with thermal and shortwave infrared imaging. He is a 2021 recipient of the NIH Director's Early Independence Award, which supports his work to develop novel infrared imaging modalities that objectively assess skin disease at both large and small scale and across diverse skin types. Dr. Smiljovic is an active collaborator with the Open Oximetry project. Thank you so much for joining us today. I'll hand it over to you.
Leo, we can't hear you.
Hello, can you hear me now?
We can hear you now, thanks.
Perfect. Okay, well, thank you guys for having me. Thank you for the speakers that preceded for their work. We've talked about, we've touched upon these questions of like, what is the best way to measure skin tone? What are different strategies for doing it? What are the different tools that exist to do it? And today I thought I would just focus in on a different question that has come up quite a bit since we had those initial discussions. The initial discussions kind of centered around using the individual typology angle, the ITA, which is derived from the cosmetics industry, but part of the ISO standard for sunscreen and likely going to be part of the standard for the new standard for pulse oximetry. But it's an established tool for estimating skin pigmentation. And I think a lot of people probably agree that measuring it at the site of the sensor is a reasonable place to go if we think that it's really light interacting with pigmentation that's causing the signal change. So at this point, I think there's several people who would say that we should use ITA, and that's a good objective measure. And then as was alluded to in the preceding talks, the Monk scale is an improvement upon the Fitzpatrick scale in the sense that it's actually a visual analog scale. There's defined colors to use, and it covers a broader range of pigmentation. So having said all that, the question that has come up is, what is the connection between these two scales? One, the Monk scale, this is a subjective scale. ITA, this is an objective measure. How do we think about them in combination? So through the work of Dr. Hendrickson and Dr. Lipnick and others who are working on the Equiax study, they shared with me data from 275 subjects for which we have the following. We have Konica Minolta CM700D spectrophotometry done at multiple skin sites, including the sensor site, as well as the forehead. And then we have a Monk scale that was determined by someone as part of the study team based on a perception of the skin tone that they see on that patient's forehead. So we have the same site being evaluated with a subjective and objective means. And we also, in all these subjects, have a Fitzpatrick scale, a perceived Fitzpatrick scale. They're not asking whether the person has a propensity to tan or burn or anything like that. It's really just how do you look on a scale of 1 to 6 using one of the many color scales that are available online that try to a sense of what a type 1 or type 6 skin should look like. So just to start with the Fitzpatrick distribution among these 275 subjects, there were 15 with type 6 Fitzpatrick skin, perceived Fitzpatrick skin, 15 with type 5, a few with type 1. And this is kind of the distribution of ITAs that we would see, again, measured at the forehead for all these different subjects. And this recapitulates things that have been published in the literature before, where you see a fairly wide spread of ITA values ranging from, the very light categories down to the tan or dark or brown categories within the same set of Fitzpatrick values. So that is not a surprise. That is consistent with what we've seen in the past. However, what I would say is, if you were looking at this data set and you said to yourself, do I have a good representation of people with dark pigmentation, you would say, wow, I have 15 subjects with dark pigmentation. That seems like a good amount. And if you were using 5 and 6 as your cutoff, then you've got 30 subjects with darker pigmentation. If you look, however, at the Monk scale, this is kind of the, I'll walk you through this. These are Monk A through J, or 1 through 10, J being the darkest, A being the lightest. And then this is the ITA values that you see for these values. It turns out actually that, and you can see the spread here, and then you can see the histogram here, that actually there are no subjects from the darkest bin of the Monk scale in this 275 subjects. So it right away tells a very different story, which is that if the goal is to capture a full spectrum of skin pigmentation, if indeed there are people out there who are a J on the Monk scale, which Ellis will talk about, and in his studies, we know that that's the case, then we are really not seeing a large number of very darkly pigmented individuals. We're seeing people with Monk scale G, we're seeing people with Monk scale F, but when it comes to H, I, J, there's 0 J, there's 2 I, and there's about 10 H. It just tells you a different story, a different picture of the kind of diversity, at least perceptually, the color that is perceived by people, the kind of diversity being included. And in terms of ITA, the range, it gives you the expected result, which is that the darker someone is perceived to be, the lower their ITA is. And certainly the ITA values get down to as low as -60 down here, which is, by any standard, if you look in the literature, whether people are looking at sun-exposed sites or sun-unexposed sites where ITA will vary, -50 is very dark. However, it would be very interesting to know what happens to this ITA range if we were to fill out this I category more, or even this J category more. So if we say to ourselves, well, as long as we get to -60, we've really covered the full spectrum of skin pigmentation. If there are a group of people who have dark pigmentation and would be characterized as J, are they -70? Are they -75? It's hard to know. I think that from what Dr. Lipnick was saying, what Mike was saying about studies in Uganda, I think it'd be very interesting and very important to understand the true range of values that one would expect, and not to be, there have been studies of ITA being done across the world, across world populations, and numbers do get fairly negative, but showing that consistently, especially in the context of Monk scale, I think would be a good addition. So this is One other way to look at this, which is kind of thinking about comparing Monk and Fitzpatrick, you can see that for each bin of the Monk scale, A, B, C, D, E, F, G, you have a certain range of Fitzpatrick values that you would assign. And it makes basic sense. People in the lightest categories are categorized in the lightest categories of Fitzpatrick, and people in darkest are in the darkest categories of Fitzpatrick. But it does show you that in some sense, by having 10 bins and having a very well-defined color scale, you are in some sense stretching out and having a higher resolution view of human pigmentation and the diversity of human pigmentation. That's what I wanted to share today. I think that with additional subjects enrolled, both at the lightest edge and the darkest edge of these scales, we will have a better sense of how much we believe these standard deviations, how much we believe these ranges, how tight these ranges are. But this, as far as I know, is the first data that I've seen connecting ITA with the Monk scale. And I'm excited for people to see this and to comment on it and to see where we go from here.
Thank you so much, Leo. Do you have a couple of minutes for questions? I know you
Absolutely,
had
yeah, I do.
Are there any questions, comments? All right, I don't see anything in the chat either. Great, thank you so much.
Can I ask a question?
Yes, please go ahead.
Keith Holloman, Medtronic. And so when we look at the ITA distribution, how do we balance that with some of the current ISO requirements in terms of going down to that -50? Do we feel good about those proposals and how we are actually going to ensure the diversity of our testing?
Yeah, I think that's a great question. I think that from my vantage point, looking at this data, it tells me that if you are screening for patients and are confident that you're finding people with a Monk scale of I or J, then you are going to be, or H, I, or J, you will be near that -50. And I think that if you get Fitzpatrick 6, you will not necessarily be near that -50. I think that the data here and the data that's been published before tells us that -50 is a real physiological amount of dark pigmentation, that exists in the world, that is not a level of pigmentation beyond reason. And I think from the perspective of ensuring that we have equity in our devices, it's important that we include people at those extremes of pigmentation in the studies. So from this perspective, I would say, if you were using Fitzpatrick as an initial screen, then you may not really reach that -50 if you got type 6 skin, because it just doesn't capture it. People who are dark and people who are not as dark are both bundled into the same group. If you were to use the Monk scale as a screening mechanism and you were getting people with a darker pigmentation at the higher levels of that Monk scale, I think you are more likely to get that -50 ITA.
Thank you.
Thanks for that question, Keith. We have a question online. Francois Lelouch, please unmute yourself and go ahead.
Yes, thank you to share this interesting data. Do you have some data with a Delphin colorimeter, because you also had this device in previous protocol, and is this the same type of figure, and do you recommend to use it in addition to Monk
Sure.
scale?
It's a great question. When I've looked at comparisons of Delphin and Konica, they give similar answers. They're not exact. I think that in terms of giving you an ITA estimate, I think they are close. And I think that both could be used. I prefer the Konica Minolta because it is a more complete instrument. It's a spectrophotometer. It's measuring light at essentially 30 different wavelengths. And the Delphin Color Catch is a tristimulus device that measures light at essentially 2 or 3. And so I think that in terms of getting you a parameter in LAB space, both will be good. I think from the perspective of doing the science, because as Phil Bickler just said on here, like the relationship between exactly, how does ITA directly predict the level of pulse oximeter error? That connection has not been made yet. And so as we're thinking about those connections, to me, having a device that gives you the full spectrum, maybe it turns out that ITA is not the best number, but some other derived parameter from that spectrophotometer reflectance curve is what we end up wanting to use. So I like the idea of using the most robust device, but I do think that others have compared the data and found that even devices less expensive than the Delphi color catch colorimeters provide very similar results in terms of a predicted LAB color space and then the subsequent derived ITA measure.
And Keith, I think Phil made a comment to your earlier. Thanks, Francois, for the comment. But Phil made a comment here. Phil Bickler saying, I think in order to fully answer, and Phil, correct me if I'm wrong, in order to fully answer that question, I think we need to better understand the relationship between ITA and device performance before we can feel it's almost two separate issues. Certainly there's room for improvement of diversity of skin color, and we're taking a best guess and seems to be reasonable. But then the only way to know for sure is to have better data on the impact and the relationship between skin color and device performance, which may or may not be at that threshold, or you don't know that relationship perfectly yet. Phil, is that?
The only other thing I wanted to mention, just importantly, this Equiox data comes from people who are not healthy necessarily subjects, right? These are people who are sick. And something that we need to keep in mind as a group is that when you make colorimetric measurements, like ITA, which is a light-based measure, right? And pulse oximetry is also a light-based measure. There may be things that impact the number that you get, which is supposedly an estimate of melanin, that may throw off that estimate. So, I have seen myself in studies with pigs. I've did pig studies and looking at the color change with congestion of a flap, creating congestion locally in the skin. You can make the color of the skin change without changing the melanin, and the ITA number changes. So it's well within reason that some of the data I showed from people who are sick, that the ITA is not a perfect surrogate for the melanin content, and therefore the connection between what we perceive as the level of pigmentation is not perfect. I think in the validation studies that are going to be done in healthy subjects, hopefully that will not be as much of an issue, but I do want to throw that caveat into the data from the Equiox studies.
All right, we'll take one more question from Wim. I think you had your hand raised, and then in the interest of time, we'll move on. So please drop your questions in the chat and you'll get us as we go.
Okay, I'll try to be quick. I fully agree with you that the focus should be on getting the darkest skin tones included rather than just a dark group. Second of all, I would like to make a remark that I think we have discussed in the past that measuring the skin color is just working on a hypothesis that indeed the skin color is the cause of these SpO2 errors. And I think at least I'm fully open to any other root cause, and that ITA is perhaps just a confounder. But I think we've said in the past that if ITA is indeed, well, I'm not going to go into the statistics. I think I've made my point clear. I just want people to realize that skin color is not the only thing that is variable in ethnicities. That's all I want.
Right. I mean, I think we've said before that, race is not a surrogate for pigmentation. And I think one of the things that all of us have mentioned as a potential issue in the Schoeding study was that it was self-identified race, that we actually don't know what the pigmentation levels were in those studies. I see some comments in the chat just asking which location. The data I showed was the location specifically from the forehead. And I think, again, just to clarify, I'm not saying that forehead pigmentation should necessarily tell us something about the pigmentation present at the sensor site, which includes both, where the light goes in and where the detector comes out. And if you're looking at the fingertip, you're talking about a sun-exposed site, which is darker on the dorsal side, and a sun non-exposed site, which is lighter on the ventral side. And so the things get complicated there quickly. I think that the one of the goals here, right, is trying to come up with a two-pronged approach, right, a two-step approach where we say, how can we be confident at the outset that we are enrolling people with a wide diversity of pigmentation and without making a measurement with a colorimeter using something like the Monk scale, which has a broad range of pigmentation, far broader than the Fitzpatrick scale, that seems to make sense. And when you look at the connection between Monk and ITA, then you at least see that the people with darker skin certainly have a lower ITA as expected. And if you go to the extremes, it seems to get to the extremes of what we think ITA to be. Beyond that, whether there's an overlap at a given Monk scale and how much ITA varies, these are open questions. I mean, this is as far as I know, this is the first time where we've been able to measure with colorimeters an objective measure and couple it and compare it to a subjective measure with the Monk scale. It's been done for Fitzpatrick. It's not been done for Monk. So this is, hot off the press. This is brand new. Thank you for your attention. And, I'm happy to talk offline as well if you have further feedback.
Thank you so much, Leo, for such an insightful presentation. Next, we have the honor of hearing from Dr. Ellis Monk. Dr. Ellis Monk is an associate professor of sociology at Harvard University. He earned his PhD in sociology from the University of California, Berkeley, and his BA in sociology from the University of Michigan, Ann Arbor. His research focuses on the comparative examination of social inequality, especially with respect to race and ethnicity in a global perspective. He is the creator of the Monk Skin Tone Scale and a 2022 recipient of the NIH Director's New Innovator Award, which support his research on race, skin tone, and pulse oximetry. Dr. Monk is also an active collaborator with the Open Oximetry Project. I will hand it over to you.
All right, thank you very much. Can everyone see my slides?
Yes.
Yes.
If you put it on presenter mode, that'd be great.
All right, 1 second. All right, thank you. So what I'll go over today is a lot of the research I've been doing with Google to validate the Monk Skin Tone Scale in the US and beyond. I'm mindful of time, so I'll try to skip through as much as I can skip through, but I do want to start off by reiterating that census categories which measure race, ethnicity are not the same characteristic as skin tone. And there's lots of research, including my own research, which shows that heterogeneity in skin tone within and across different ethno-racial categories around the world are associated with inequalities in education, earnings, and in a lot of my research, I show this in mental and physical health disparities. And one of the mechanisms that people propose is discrimination. So I think it's important, at the beginning of this to say that we still have a lot to learn about how skin tone is related to pulse oximetry. And it may be the case that there are also social determinants of health that are playing a role in all of this. And that's where having an objective and subjective measure to capture those human perceptions, I think, is really vitally important as we begin to do more research in this space. So there's lots of stuff that's come out in the last few years about what people refer to as colorblind technology. This isn't just pulse oximeters. This is stuff like self-driving cars, search algorithms on Google, Instagram, all kinds of things. But today, talking about these pulse oximeter regulations, as was already said, we know that the standard, currently is 2 subjects or 15% of the pool. And the question becomes, who is darkly pigmented? And certainly objective measures is one way to do that. Historically, people have also used this thing, the Fitzpatrick scale, which I think we're all well aware of. I'll just say one thing, which is that as Leo showed, one common criticism of the Fitzpatrick scale has been that it doesn't do a good job of capturing the dynamic range of different skin tones in the human population, especially when we take darker skin tones into account. And one thing that's also been clear in my research with Google, validating the scale and working with the scale, is that the Fitzpatrick scale also has the strange property of truncating at the lighter end of the scale as well. It kind of sits in this middle range of different light brown to somewhat darker brown. So there may be this kind of dual problem of not being light enough and also not being dark enough simultaneously. And this has led to a lot of criticism, and this is Homeland Security talking about rethinking their use of the Fitzpatrick scale. So I designed the Monk scale some years ago. Here's a picture of the swatches, which are, one way of using it. There's also the orbs, and we've been doing testing at Google to see if there are comparative advantages between the swatches and the orbs. And I think most of you are all, kind of up to date on the Monk scale, but I validated it initially with the NSHAP project, which is a gold standard health focused survey, which has received over $50 million in funding from the NIH and NIA. And we did cognitive interviewing and all kinds of other testing. And NSHAP is out in the field right now collecting data, and they use the Monk skin tone scale to collect data on skin tone. So last year, Google announced that they launched the Monk skin tone scale. They've adopted across all kinds of different products, from testing their Fitbit to improving skin tone representation in the Pixel camera phone. They've actually started to do work embedding it in the search engine, the image search engine as well. And a lot of the decision making around that was based on the research that we've been doing, validating the scale that is now on the cusp of acceptance and also being accepted. So first of all, there's this study, which is just about to be accepted, should be out early next year in ACM journal on responsible computing, where we wanted to look at the extent to which, using a nationally representative sample of people from the United States, how people perceive the representativeness of the Monk scale compared to, say, a larger scale, 40-point scale from the cosmetic industry, and also the Fitzpatrick scale. This study showed that in the US, the Monk scale is perceived as just as inclusive as this 40-point scale, and the Monk scale is only 10 points, and significantly more inclusive than the Fitzpatrick. And this holds across the entire skin tone continuum, especially for people with darker skin. We've also begun to do a lot of work looking at global validation of the scale, both in terms of annotation and perceived inclusivity. And this is all very, very large scale research using nationally representative samples in all of the countries I've listed. And there's actually even more countries involved in this. And we're asking questions around what is the level of consensus using the scale among lay people, but also trained experts. How many annotators should we use per subject? How does annotator consensus vary across different regions in the world? And we're also asking these inclusivity questions, which are vitally important for diversity and equity reasons all around the world. And on top of that, using these questions that we have about usability, because another concern, of course, is that you want a scale that is representative and equitable, but you want people to be able to use it. So that's another aspect of our global validation testing. We have a paper that was just accepted at NeurIPS, which is a leading computer science conference, and we're very happy that this is going to be out pretty soon. And this is our first global validation survey where we tested the Monk scale in relative to the Fitzpatrick scale. Using the Monk skin tone scale, we were able to ask people all around the world, annotators who are both not trained and experts in dermatology, to use the scale and to try to see the level of consensus using a data set of photos of different human beings in well-lit conditions and in poorly lit conditions as well. And what we find is that using that plus or minus 1 threshold of discrepancy, we find that in our pool, our global pool, the consistency with my own personal annotations and even annotations across regions, plus or minus 1%, is somewhere around 95% consensus. And the average distance, varies, but it tops out around 0.78, which is to say that usually the ICCs that we see using this global approach to annotation with pools of annotators all around the world, we find that the ICCs range from 90 to 96%. Now, of course, there's going to be some overlap because of the plus or minus 1 threshold that typically is used with these subjective scales. But as you can see here, even a poorly lit image, and we're comparing raters from India to raters in the US, we still have overlapping distributions in how they view these particular images. So as I point out, here's just, from the paper itself, you can see at the bottom the golden images, which are like the well-lit images. The ICC range from 0.9 to 0.96. Using the challenging images, it ranges from 0.82 to 0.95, but it's still pretty consistent. And the challenging images are extremely poorly lit, so I don't think they're that relevant for this particular work on looking at pulse ox validation. And what we find is that skin tone can be reliably annotated using the MST. It doesn't matter if people are trained or not. Usually we recommend that you have at least 2 annotators so that you can, or 2 to 3 annotators. Usually statistically 3 is better than 2, but if you can have 2, that's fine as well. What we're seeing in even more extensive research, which is still underway, again, always using nationally representative samples around the world, is that even when we just use 1 annotator and we have like a nice pool, we get pretty clear consistencies all around the world using the MST, plus or minus 1 point on the scale. And in this particular use case, if we think about coarsening the MST to focus on 8 through 10, or HIJ to determine dark, because of the consensus that we see plus or minus 1 point, that means that you can be very confident that if you're getting someone in that dark category, plus or minus 1 consensus is so strong that you're probably going to get someone that's very dark skinned. And this is great for recruiting and making sure that we have great coverage across the entire skin tone continuum, as Leo pointed out. And some of the usability research that we did, I mean, I can go through this pretty quickly just to hit some key points. But one of the things that we saw is that the Fitzpatrick scale, people like to use it because it's only 6 points, but we find that the Monk scale is just as easy to use and offers more options, especially for people who are darker skinned. And in particular, it has much higher levels of perceived inclusivity all around the world compared to the Fitzpatrick, and even a scale that's 4 times larger from the cosmetic industry. And importantly, we also found in new testing that the Monk scale also includes undertones, which matches the skin tone of the respondents that we have sampled from around the world as well. At a larger scale, I know some people, one thing has come up thinking about even larger scales, potentially like, well, how can we do, better in terms of covering the skin tone continuum? But one of the things that we notice in our research at Google is that there are some diminishing returns where if you go beyond a certain length of the scale, the consensus starts to drop precipitously and people just aren't able to reliably use a larger scale. So we pretty much stick around 10 points. I think I pointed out before that it's been adopted by Meta or Facebook for benchmarking fairness in data sets and video. And even more recently, they've adopted it in a new data set on images called Facet. They're also using the Monk scale to do that for computer vision and images as well. Again, they did their own independent testing of Google using the Monk scale and putting this benchmarking data set together. And then some developments with Pantone. So for the past several months, I've been working with people at Pantone. One thing to highlight here is that there's actually 2 different systems. So on the one hand, there's Pantone's color system, which has their Pantone matching system, which you see used in interior design and fashion, and even the Pantone skin tone guide. And the Monk scale has actually been matched with Pantone already. And I've seen some mock-ups working with them on kind of perfecting the standardized printing of the Monk scale. But we're also beginning to explore a Munsell style product for the Monk scale, which is a higher level of standardization, which we've seen used in soil samples, rock samples, and beads. This is an even longer and more drawn out process of standardization, which involves moving from the digital realm to like physically mixing paints and all kinds of calibration techniques. But in working with them, one thing I learned is that the HQ prints that people can kind of use, just kind of downloading the Monk scale online, should suffice for now until the Pantone prints are ready, mainly because the Monk scale, the swatch version in particular, is already standardized. So most printers should be able to reliably print that out in high quality settings. So this is still work in progress. We're getting a little closer. I've already seen some mock-ups, but I just want people to know this is on the horizon. So some concluding thoughts here really quickly. The findings show that the Monk scale is easy to use as the Fitzpatrick while being significantly more inclusive and equitable. There are high levels of consensus using the scale, plus or minus 1 point on the scale. And if we coarsen across the scale, that means that we'll see even more consistency if we just break out into regions of light, medium, and dark on the scale. The Monk scale then may be used to recruit a diverse pool of subjects across the skin tone continuum, as is done similarly in artificial intelligence and machine learning data set benchmarking. And one thing I really want to highlight here is that the subjective and objective measures of skin tone are both important to collect because they may help tap into different mechanisms through which skin tone may be implicated in pulse oximetry issues. And there's so much more to learn about the role of skin tone in pulse oximetry. I think this is a massive opportunity for us to scientifically characterize what the different contributions are between the social world and the biological and physiological aspects at work. And there's one added bonus, which is that the subjective measures are also good at communicating to the world who was included in these studies. I don't have it on this slide, but Leo has shown this before. If you look at visual outputs from devices, they don't usually match how human beings view their skin tone or the skin tone of others. They tend to look very, very dark. They don't quite proxy one-to-one how human beings see skin tone. So the other aspect of using a subjective measure here is in communicating to the public, hey, we've crossed this range of different skin tones to include people into these studies. And what Leo showed today is that the 8 through 10 range on the Monk scale is getting you down -50 and even lower in ITA. And that's something that the Fitzpatrick scale simply is not doing. So I'll stop there. Thank you.
Thank you so much, Ellis. I think we can take a couple of minutes for questions if there's, yeah, I see a hand raised, Francois, please go ahead.
Yes, thank you, Dr. Monk. I think it's necessary to introduce these new scales in evaluation, but I have a practical question because as we evaluate patients, it may be difficult to have always the same light patient which with near the window or a patient with other artificial light. Did you see a difference when you evaluate with different type of light, a difference with your scale? Because it's clear that with colorimetric method there will be less impact probably, but it's impossible to imagine that all study will be conducted with colorimetric methods.
Sure. So the closest we got to replicating that was with the challenging images data set in the paper that's accepted at NeurIPS, where we altered the lighting conditions on the photos. And certainly you do see a distribution shift, slightly darker and darker lighting. But the overall, like if we were to look at the well-lit image and the one that's poorly lit, there's still an overlap in the distribution of the subjective perceptions of the skin tone of the person in the photo, all of which is to say that human beings are adjusting for light. So they are capable of doing that. So of course, you would think in practice that a colorimeter would probably do a better job. But I want to give us humans some credit here. We also are able to adjust quite well for poor lighting and looking at skin tone.
Thanks, Alice. Are there any other questions in the room or?
Well, perhaps, yeah. I was interested in the remark you made about the Monk skin tone scale matched with the Pantone skin tone guide. Does that suggest that we could take some swatches from the Pantone skin tone guide and compare those with Monk skin tone grades? Because that would mean that there's not so much a need for, or we can start using the Pantone skin tone guide already as a factory printed guide.
So let me be clear. So the Monk scale has been matched within Pantone's system, but that doesn't necessarily correlate to their skin tone guide. So they're not one-to-one. So what they published in their skin tone guide doesn't necessarily totally match up with the Monk skin tone scale. The skin tone guide is really extensive, and some of their colors don't actually even perfectly match the Monk skin tone scale. So it's in their system, but it's not in their skin tone guide per se.
All right, I see one other question in the chat. Dr. Hendricks, do you want to unmute and share? Or?
Sure, this is for Dr. Monk. Dr. Monk, while MST is not a proxy for race and ethnicity, have you investigated how MST may predict race and ethnicity?
So I think the way I would look at that is to think about the skin tone distributions within and across different ethno-racial categories. And how that works, at least in the US. And of course, once we think globally, this becomes extremely messy because of the number and the differences of the census categories used all around the world. But if we just think about the United States, it is certainly the case that like in the category Black, the distribution of MST values is different than in the category, say, white. But things get complicated right away because you have in the United States the practice of marking one or more categories on the census. We also have Hispanic and then self-identified white Hispanic, Black Hispanic, and then Black, white, whatever mix you want. So once you add in all of those complexities and you go beyond just kind of the simple kind of ethno-racial binary of Black, white, once you get beyond that, the skin tone distributions and the variance in skin tone gets very, very crazy. So to a certain extent, you can predict. But when you actually think about the great diversity in the United States, the skin tone census category relationship, you kind of have to throw it out of the window.
Thank you so much.
Thanks, Ellis. Thanks, Leo. Thanks for the questions. We're going to move to the next and final section of this, which will go, we're a little bit behind schedule, so we're going to probably get pretty close to the noon hour. We'll regroup at noon about how much of a break we'll take between the next session. Sorry about that for those who are joining that. Dr. Hendrickson, if you want to start sharing your screen, I'll start introducing you. Thanks for joining us. We know you're on service in the ICU and are spending some time with us this morning, so thanks for doing that. Dr. Hendrickson, for those of you who haven't met, is a pulmonologist, critical care specialist at San Francisco General Hospital. She conducts multiple clinical trials on acute lung injury and respiratory distress. She's also the medical director of the medical intensive care unit here at San Francisco General Hospital. She is one of the co-leads for the FDA CIRSE-funded Equiox study. Thank you again for taking the time. So without further ado, over to you.
Recorded now. Hi, everyone. Thanks for having me. As Mike said, I'm on service right now in the ICU, and I hope to be available for the duration of this short presentation, but may need to step away. I wanted to give you all an update on the Equiox study, which is the actively enrolling prospective study of pulse oximeter errors across a range of skin pigmentation in patients in the intensive care unit at San Francisco General Hospital. So our enrollment update is that we have 352 fully consented patients enrolled with 693 arterial blood gas samples collected. These are observed by the research study team. And so we have direct observation of the comparative SpO2 reading, as well as the quality of that tracing and the stability of the pulse oximeter reading from our standard of care monitors, which are Massimo and Philips. We have a few additional patients who have been enrolled but are not fully consented yet or are on surrogate consent. And we are actively recruiting and ongoing enrollment in this study. I don't need to repeat what Dr. Monk said, that Fitzpatrick scale is limited and probably not the best or the most scientific approach to categorizing skin tone. And I'm also going to show you Van Lushen scale distributions. I'm not presenting those to suggest that they are adequate, but simply part of our data collection and to give you kind of a rough idea of the distribution of skin pigmentation in this cohort. As you are well aware, the skin pigment assessment differs across anatomic sites on the same person. And so we collect data at several different sites, and I'm presenting to you data collected from the dorsal DIP because most of our pulse oximeter readings are from oximeter probes on the finger. We're also collecting Monk skin tone scale for these subjects. And you can see that we have a decent representation among the mid skin tone ranges on this scale, but have pretty sparse representation in the very darkest categories of Monk skin tone scale in our population at San Francisco General. I will say, just because this question comes up a lot, we are enrolling everyone. And so we approach all patients and offer participation if there's an arterial line in place. And so we're not selectively recruiting based on anything besides if you have an art line or not. And I think this limitation that we have is a reflection more of the patient population that we serve at our hospital rather than any kind of selection happening at the study enrollment and recruitment level. You can see that our pulse oximeters are mostly on fingers, but we do have quite a few on earlobes. We're collecting skin tone data at the face site and also attempting to collect spectrophotometer data at the earlobe, but it's pretty hard to get those measurements on a small piece of skin that's very floppy like the earlobe. So we have some practical challenges with some of the earlobe pulse oximeter readings. And I will also say in practice, the folks who get an earlobe pulse oximeter, it's usually because we're having trouble picking up a reading on the fingers. We have a pretty diverse population as assessed by standard self-identified and charted race categories at San Francisco General. I will say you can see we have quite a large number of patients enrolled who identify as other. The vast majority of that group identifies as Latinx ethnicity, but there is, of course, a little bit more variation to it than that. So we have, this is our ethnicity data. Again, this is just kind of what EPIC, the electronic medical record, captures for self-identified race and ethnicity. Here is a scatter plot of our raw data comparing SpO2 on the x-axis and SaO2 on the y-axis. And you can see that the vast majority of our observations are above the 90% saturation rate. The color coding of these dots shows that most of the samples we observe are in stable pulse oximeter readings. So projecting the same estimate of the oxygen saturation for 30 seconds or more. Those are the green dots. The red dots show you when that tracing is not stable for 30 seconds. This has been a challenge for us. One that maybe we should have anticipated because clinicians rescue patients when their stats are in the low 90s or high 80s. So it is just not something that's tolerated by the nursing staff, the respiratory therapists, or the physicians to have someone kind of in marginal oxygen saturations. So we do have it's pretty rare to find someone who's satting less than 90% in clinical practice. And this is consistent with other published data as well as some surveys we've done with collaborators at other institutions. We have about 12% of our observations reading in the less than or equal to 90% SpO2 range. This is just a frequency distribution of that same scatter plot data, just showing that everything is very right shifted. So we plan to continue enrollment for the next 6 to 12 months. We have explored some ideas for site expansion. There are no current plans for that, but we're actively looking to collaborate with people doing similar work because the study would be more generalizable and stronger if we had data from other institutions as well. We would love to collaborate with anybody doing this work and have our protocols available for open sharing and are available to help anyone starting research collection or data collection, or people who might be willing to add their data to a group repository that would make for better science. Next steps also include adding additional study oximeters beyond the standard of care monitors that we're currently using right now. So with that, I'm done with my presentation and would be happy to answer some questions.
Thanks, Carolyn, for the presentation. Since you are on service, before we get to Dr. Conrad's presentation, maybe see if any questions from anybody in the group.
I posted one.
Go ahead, Julian.
Hi, Julian Goldman here. Just wanted to clarify your definition of stability at 30-second period. I just posted in the chat. Is it an unchanging displayed saturation value, or is it plus or minus 1%? How do you define stable?
Yeah, so it's not unchanging, but we allow for a variation of plus or minus 1% in the readings.
Okay, thanks.
And then Julie, there are some, we would look through some other protocols of like recent suctioning, recent ventilatory vent changes, recent oxygens. We do try and pay attention to those things and are talking about how to spell that out a little bit more clearly to standardize across studies. There has been some heterogeneity, but we generally try to, as Dr. Henderson said, have a CRC there, make sure that things are not changing and that the SAT is within 1%.
You could add something like no change greater than 3% in the preceding X number of minutes as well. Like that would eliminate the restabilization or suction or something that's potentially add some complexity, but it'll ensure greater stability.
Yeah, fair point. Steve.
Sure. So I want to ask the same question for a perfusion metric. I know that you're using devices from different manufacturers that have different criteria for their perfusion metrics, but are you able to record or have any data on the stability of that during your readings?
Yeah, thanks for that question. We record the perfusion index that's projected on a monitor at the time the blood gas is drawn, but we have not been recording stability over time in that projected value.
Thank you.
Any final questions? I see a comment from Brady at Rush. Thanks for the update. Got your Konica device en route. We're excited to see your data from your prospective study coming out and contributing to the discussion. So thanks for sharing that. Any other comments or updates before we go to Dr. Conrad?
One quick question.
Please, Keith.
Because of the stability of the saturation levels, and you mentioned that typically physicians intervene in the low 90s. It doesn't get much lower than that. What other scenarios, what I would call extreme situations, are you seeing where you do have the opportunity to assess saturation at lower levels prior to intervention?
Yeah, so I think the biggest opportunity there is for patients who have acute respiratory distress syndrome, ARDS, because the protocols manage these patients with goal SpO2s of 88 to 92%. So we're about to embark on another respiratory viral season. We're actually already there. And so we'll see more patients with pneumonia and ARDS, and they should stably be in the low 80s, high 80s, low 90s. We do not have many patients with congenital heart disease. We do not have patients who are receiving extracorporeal membrane oxygenation therapy or other mechanical circulatory support at our hospital. So that's the major population that would contribute data in more stably low saturation ranges.
Thank you.
Thanks, Dr. Hendrickson. Francois, if you have a quick question, over to you.
Yeah. First, I want to congratulate you because I know how difficult it is to enroll this type of patients. It's clear that healthy subject studies are necessary, especially for desaturation, because it's extremely difficult to imagine to have enough patients with large desaturation, and nobody wants to let the patient desaturation. Everybody increase the FiO2 immediately. So it's, I think, not possible to have data other than usual data. And this is probably 99% of the value that clinicians have. So I think it's very also important to know exactly the bias of different devices. So, we conducted quite similar study, but with different oximeters concomitantly. And I think that's why that was my comment in the chat, that there's data reflect on the real gap between pulse oximeter and saturation, because in healthy subject study, probably like you wrote for skin pigmentation, the gap that you see, the bias that you see, is probably reduced in comparison with what we see in patients. So probably the two type of study are needed. That's why I was curious to know if the ISO will modify and recommend also to get some real life or patients data rather than only healthy subjects. Yeah.
Thanks for the comment, Francois. With that, I think we'll move on to Dr. Conrad's presentation, and we will wrap up the session. Dr. Conrad, I'll invite you to share your slides while I introduce you. Dr. Conrad is an internist and pediatrician with a passion for population health and health disparities, also chief pediatric cardiology fellow at Stanford. Her research interests include health disparities, and she's co-investigator on the FDA-funded study investigating accuracy of pulse ox in children with different levels of skin pigmentation, which she's going to share some updates on. Thanks for joining us. Over to you. And we can't hear you yet if you are speaking. I think you're on mute.
There we go. Sorry about that, guys. Can you hear me now?
We can hear you now, and if you could enter presentation mode, then we're looking all good. Thank you.
Great. Okay, so I'll be presenting from a pediatric perspective, just to let everyone know that we are FDA funded for this trial, as detailed here. So quick, just kind of agenda of what we'll go over briefly for this presentation, and I'll try to get through it pretty quickly. This is our team here. That includes a combination of everyone from UCSF, CERFC, Stanford, our data monitoring committee, as well as our FDA. Again, this is our team here at Stanford. Since the last time we presented, we have added some new faces. We have a second research coordinator, Brian, as well as we've added Dr. Michelle Williams to help us with our minority recruitment. So this is kind of our study timeline. We started back in June of 2022, and now to date, we kind of did complete our initial sample size enrollment in July of 154 subjects, but after our data monitoring committee met, we've had to extend our enrollment sample size. And so we've received IRB approval for that, and we're in the process of bringing on additional centers to help increase our enrollment, and we'll complete a total enrollment sample size of 312. So far, our results to date, we have recruited about 183 subjects. As you can see, we have a almost equal spectrum of less than 2 to 12, and then 12 to 22. We have a big majority of female patients. We also have a Hispanic, Latino ethnicity included. And then we have a diverse spectrum as well. And similarly to the adult study, a lot of people that do click other for race happen to be Latino in ours as well. So this is our data thus far. We've included 183 patients as before. We've gotten 692 samples thus far, which is a 90% rate. Our goal is to achieve about 5 samples per patient, where our average is about maybe 3.8 across all. As you can see, we're kind of heavily more skewed towards a lighter skin for using the von Luschen dorsal index finger. We are slowly trying to include more in our dark, medium, and dark categories. To right now, with the von Luschen's, which is on that scale of 29 to 36, we have about 8 patients there. According to the Monk scale as well, we have patients 3 recruited in G, 4 in H, and 1 in I, none in J. And as you can see, our spectrum of that. And then this is our correlation plots of our SaO2 to SpO2. And again, this is just more of our similar things in showing like our samplings of paired SaO2 to SpO2s based off of that. And as you can see, we're trying to recruit more in this darker range here. So for our statistical analysis, we've initially had a sample size calculation of 154, and then our DMC met and recommended increasing the sample size to 312 to include a minimum of recruitment of at least 20 von Luschen category 4 patients. So some of these things we went over before in prior meetings, but there's always a change in technology. So we did work on getting the Massimo 7, Massimo Rad 7 device rolled out through our center, and that's what we are testing. We learned that there's no standardized skin pigmentation scale, so we are using the Monk, the Fitzpatrick, as well as the von Luschen. And preterm infants are eligible infants to ineligibility criteria due to the risk of hypothermia outside of the isolate. So sometimes it's hard to get a good accurate of that. Also with newborns and neonates, they do have a rapid change of skin pigmentation over time as well. Also reduce the impact of the pre-analytical factors of the SaO2. So we have now a portable blood gas monitor that helps minimize the time between the sample drawn and the sample actually being processed. So it's done right there on the spot. There is also dynamic changes in oxygen saturations in the operating room. So we had to anticipate significant enrollment, and we shifted enrollment from the OR to the CVICU as well as our cath lab. And that's where we've been recruiting a lot of our patients as well. Parent anxiety about how much blood is being drawn. So we've had to use additional props to kind of tell parents how much is being drawn and minimize that as well, and getting that into recruitment. And also just being strategic in when we're recruiting our parents into the study as well. Also, other things is childhood anxiety. And then sometimes when appropriate, we try to remove as much like intimidating PPE or other things that may scare children. And we've been involving child life as well as trying to minimize the fear of this study. Because as you can see how small a child's hand is, how big these devices sometimes can. They can be a little bit intimidating to our younger patients. And finally, earlier our enrollment was very much so skewed towards patients with lighter skin pigmentation. So we've changed our recruitment strategy from taking all comers to oversampling among those with darker skin pigmentation and prioritizing even those with darker skin pigmentation and lower SpO2s and SaO2s. We've examined our consent failures. We attempted to increase race minority concordance with consent. We brought on Dr. Michelle Williams as an expert in minority recruitment for clinical trials, and we developed a recruitment brochure, and we're also expanding to a multi-center trial. So our future plans right now, we will be adding Emory and CHOA as a site to the Stanford IRB. We're preparing a sub-award for them, and we're going to train Emory and CHOA on the staff and the equipment, and then begin enrollment at Emory and CHOA sometime soon this fall. So in conclusion for us, we've learned so far that protocols from adult trials cannot be directly transferred to pediatric populations. This population presents new challenges, and we've modified our protocol accordingly. We've learned a lot as we continue to enroll patients, and we have to amend the protocol accordingly to respond to these challenges. And we have doubled down on our efforts to recruit and increase enrollment amongst our darker pigmented patients. And despite all these challenges, we have enrolled 183, and we're on track to hopefully increase our recruitment amongst von Luschen 4 patients, and we're hoping to hit our enrollment target by January of 2024. So the other things I think just to briefly mention too in conclusion is that what is unique to our study and not so much to the adult studies is that we are able to have naturally desaturated children because we do have congenital heart disease. So sometimes we do have SATs, and I can even roll back up to our data slides, that tend to be at baseline a lot lower than what are expected in our adult studies. So I think that that's provides a lot of utility towards this project. And with that, I will thank you and open it up to any questions.
Thanks very much, Dr. Conrad. Any questions? Just looking through the chat. Sandy, you had your hand raised at the end of Dr. Hendrickson's presentation. Sorry for missing that. Did you still have a comment?
I just wanted to confirm my observation. It looked like most of the points in Dr. Hendrickson's study, the SaO2 was higher than the SpO2, which I think is the opposite of the occult hypoxemia that people have observed and seen. So it was just a question, am I looking at this improperly, or maybe that's what it's actually showing? And I understand there's not a lot of points there and lots of work still needs to be done.
I'm sorry, do you want me to pull up the data table again? Can you repeat your question one more time? Sorry about that.
So if that the data curve that you showed where you had all the points, and they're above 90%, it looked to me, just eyeballing it, that there was a positive bias in SaO2 higher than SpO2. Right? I mean, and that to me is the opposite of a cold hypoxemia, right? Where SpO2 is reading high and says you don't need additional oxygen therapy because the SaO2 is lower. But this was a question for Dr. Hendrickson. That was in her data. It's just an observation. We don't really need to discuss it or comment on it. We can wait further.
Okay, thanks, Sandy. I don't know if Dr. Hendrickson has dropped off. She may have had to go back to clinical duties, but we can continue that conversation offline.
I have to jump off. I got to go start the ISO meeting too. So thank you, everybody. Appreciate your participation and help.
So we're going to go ahead and conclude at this point. I want to thank everybody for joining. Also want to reiterate that we're going to, again, invite everybody who's not already participating in the Collaborators Forum, which we intend to formalize into a collaborative community along with FDA and other partners. Please sign up through the website. We're really hoping to make sure that we have as much involvement in that as possible, and look forward to continuing to work with the ISO committee, the FDA, and others on these projects. Thank you very much to all of the speakers. Thanks to those who have been able to join in person, and stay tuned for a next meeting and some of the subgroup meetings, which we'll be sending out soon. The video for this meeting and the minutes will be posted online. And for those who are sticking around for the ISO meeting, it is the same room, different link. So you should have received that at this point. Given the timing, we ran over, apologies. We'll break. Everybody get food and return as soon as possible to this room for that meeting to begin. Thank you again to everybody, and please reach out if any questions or comments.